The topic that we'll be dealing is Neisseria. The genus Neisseria contains two important pathological group of organisms, Neisseria gonorrhea and Neisseria meningitis. These organisms are gram-negative, aerobic, non-sporing and non-motile and oxidase positive organisms, usually occurring in pairs which are diplococci. And the most important characteristic feature of this organism is it is oxidase positive. And with the help of this, the organism can be easily identified. And the pathogenic group of organism is Neisseria gonorrhea, Neisseria meningitis, and the organism which is a commensal of the human respiratory tract or the mouth is Neisseria lactamica. Coming to Neisseria meningitis, it's also called as meningococcus, Diplococcus intracellularis, meningococci or meningitis. Here, this organism was first isolated by Wish Selbaum in 1887, and the organism is the one that is related to the pathogenic disease meningococcal meningitis, and was first seen in the cerebrospinal fluid because it is associated with meningitis. The organism can be easily isolated in the cerebrospinal fluid. This organism has the ability to cause sporadic disease which are in the form of localized outbreaks, or can also called as causes epidemics, that is septicemia. Coming to the morphology of the organism, these organisms are gram-negative. That is, they have a special layer which is called as a lipopolysaccharide layer that gives its virulence capacity or the ability to cause the infection. They are oval or spherical in shape, measuring about 0.6 to 0.8 micrometer, and occurring in pairs with a flattened adjacent slide which are flattened in nature. And they show variation in the size, shape, and morphology due to the autolysis or in the old culture, but in a fresh culture they show a regular shape, and these organisms which have been isolated from the lesion are mostly the one that shows the regular shapes. These organisms are non-motile, and fresh colonies or fresh isolates can also exhibit capsular material. Coming to the cultural characteristics, how these organisms shows their characteristic appearances on different cultures or the medias. These organisms are fastidious organisms which are exact in their nutritional requirement. They do not grow in a normal media, but the media has to be enriched with either the blood, serum, or the other ingredients so that they can enhance the growth of this particular organism. And in this particular growth media, the most characteristic feature of this growth media is that it inhibits or neutralizes the inhibiting substances and does not provide the extra ingredients for the organism, but it inhibits or neutralizes the inhibiting substances that allows the growth of this organism. So the media is allowing the growth of the organism by inhibiting or neutralizing the inhibiting substances and not providing any excess or special nutrients to the microorganism. This is the characteristic feature of the growth media that helps to grow the Neisseria species or the genus on a culture media, and hence enriched media is only used to grow this organism. These organisms are strict aerobes, the temperature or the optimum temperature being 35 to 37 degrees centigrade. These organisms do not grow less than 30 degrees centigrade, and the pH requirement is around 7.4 to 7.6, and they very well grow on a media containing 5 to 10% carbon dioxide with a humidity. So this has to be kept in mind when we want to isolate the organism on an artificial media. On a solid media, the appearance of this organism is small, translucent, they are round, convex with a bluish grey appearance, and they are smooth glistening surfaces, and they show entire edges. So this is the another characteristic feature of the organism which shows entire edges on a colony, and the shape is lenticular, and they are buttressed and they are easily emulsifiable. They show weak hemolysis on blood agar, that is, they could not cause the lysis of the RBCs, and they also shows variations similar to that of the pneumococcus, that is a smooth and rough variation colonies can be seen on the media, and on a liquid media they show poor growth, that is a granular turbidity is seen in the liquid media. They do not very well grow on a liquid media. They require a solid media for their growth. Now coming to the media on which they can satisfactorily be grown are the blood agar, the chocolate actinomycete agar, Muller-Hilton starch casein hydrolysate agar, and the selective media for them is a modified Thayer-Martin media, which is actually contains antibiotics such as nystatin, colistin, vancomycin to inhibit the other organism and allows the growth of only the Neisseria meningitis organisms. Next, coming to the biochemical reactions, these organisms can be identified easily with the help of certain biochemical properties. They are catalase positive, oxidase positive, and they can be grown on a subculture once they are detected as oxidase positive. In the oxidase positive test, what we do is we take 1% of the oxidase reagent, tetramethyl diamine, tetramethyl paraphenylene diamine hydrochloride. It is an oxidase reagent. When this 1% of the solution is added onto the liquid media or onto the organism, they usually color themselves as deep purple. So once we identify the deep purple colonies, immediately they have to be subcultured. Otherwise, the organism will lose its growth on prolonged incubation. And this test can also be carried out with another method which is called as a COVAX method, where we make use of a filter paper disc that has been moistened with the oxidase reagent, and the organism is usually taken with the help of a loop and then smeared onto the filter paper disc. The presence of deep blue or purple color indicates the oxidase positive reagent. So once the organism is said to be oxidase positive, immediately it has to be subcultured for the further growth of the organism. And these organisms are indole negative, hydrogen sulfide negative. They do not reduce nitrates, and they can utilize glucose and maltose. The difference between Neisseria meningitis and Neisseria gonorrhea is gonococci can only acidify glucose but not maltose. This Neisseria meningitis can utilize glucose and maltose but not sucrose and lactose, and they produce acid but without gas. So they produce acid but it is weak in nature, and they can be very well identified on a media containing peptone, serum, agar slope with certain kind of sugars and an indicator that can be added to identify the growth of the organism. Coming to the antigenic properties by which the organism is able to cause infection and is the one causing meningitis in the human. So these organisms antigenic characteristic is the presence of a capsular material that helps to cause the infection in the human host. So based upon the capsular material, this can be typed into 13 serogroups, and among which A, B, C are the one that are involved in the causation of the disease. The group A is the one that is involved in epidemics. Group C is the one that is involved in localized outbreaks, whereas B is the one that can cause both epidemics as well as localized outbreaks. And the other group that causes the meningitis is group 29E, W135 and Y. So all these 6 groups are the one that are related to the causation of the disease such as meningitis. Coming to the resistance of this organism, these are highly delicate organisms. They cannot survive without the host, and they immediately die when they are exposed to the external environment. Heat, they are heat sensitive. They can be easily killed by desiccation, that is a removal of the water. Any alteration in the pH condition can also affect the survivability of this organism, and they are easily killed by minute doses of antibiotics. But recently it has been found that there are certain resistant strains that has come up to cause the infection. Coming to the pathogenicity, how this organism causes infections in the humans, the 2 type of diseases that are caused by this organism are cerebrospinal meningitis and meningococcal septicemia. Usually the organism harbors in the nasopharynx, and they are the strict human parasite of the nasopharynx, and asymptomatic carriers can carry this organism, and sometimes the organism causes infection in asymptomatic carriages when the immune system has come down. Otherwise they are only the carriers, and they can transmit the infection to the other person. And the local infections, dissemination of the local is very poor, and sometimes it is ensured via the rhinitis or the pharyngitis. Coming to the spread of the infection from the nasopharynx to the meninges, it can be directly via the perineural sheath of the olfactory nerve, or via the cribriform sheath to the subarachnoid space to the meninges, or can be directly via the bloodstream, that is nothing but the bacteremia. Apart from this, transmission of infection from the nasopharynx to the meninges, this can also be brought about by via the conjunctiva, where they are responsible for causing conjunctival or meningococcal purulent conjunctivitis. Once they get established in the cerebrospinal fluid or the CNS system, they form the lesions involving the spinal cord, the brain, the base of the brain and the cortex. And these organisms are mostly seen in the spinal fluid, either freely or within the leukocytes, and they are the one that causes a sequel of infection leading to blindness, deafness, or can also involve the multi-system failure. Coming to meningococcemia, that is the symptoms associated with meningococcemia. It results in acute fever, chills, malaise, prostration, and the typical characteristic feature of this organism is that it causes petechial lesions. The organism can be identified from the petechial region by variety of techniques, that is culture or smear, or you can also go for the serological techniques. And it involves the metastatic involvements of the joints, ears, eyes, and the other body parts leading to arthritis, etc. And 10% of the cases it can lead to pneumonia. Now, few of the organisms can also cause fulminating meningococci, which is the secondary stage of this disease. So, initially it can cause the meningococcemia or the meningococcal infections. Next, it can lead to fulminating meningococcemia, which leads to shock, intravascular coagulation, and multi-system disorder, which is called as Waterhouse-Frederick syndrome. And very rarely it can lead to chronic meningococcemia, which this particular infections or the pathogenesis is because of the deficiency in the terminal complement proteins, that is from C5 to C9. Complement system, as we know, it is an active system that helps in elimination of the organism. So if the person is deficient in this complement protein, he is more prone to the infection from the meningococcemia. Now, the next important virulent characteristic feature is the endotoxin, that is the lipopolysaccharide layer. The organism has ability to undergo autolysis and release this lipopolysaccharide. When this lipopolysaccharide goes and interacts with the vascular endothelium, the vascular endothelium is highly sensitive and cause the inflammatory response to begin. And this inflammatory cascade system response, as well as the release of the cytokines and the nitric oxide, helps to enhance more amount of inflammatory mediators to be released, thereby damaging the host and leading to the pathogenesis of this particular disease. So ultimately leading to adrenal hemorrhage as well as shock. Coming to the epidemiology, it is the strict parasite of the human nasopharynx, and hence the organism can be transmitted via the carrier or via the patient. So in the carriers it does not cause infection, but they are more prone to transmit the infection to the other person. And the transmission is usually via the airborne droplets. No fomites or other transmission is seen transmitting this particular infection, and mostly seen in a close communities or semi-close communities where the infection can be most prevalent. And this infection can be caused to the children from the 3 months to 5 years of age, and dissemination is again in a crowded place. Coming to the lab diagnosis, how do we identify this organism in a laboratory diagnosis? The agent is the meningococci or the pneumococci or Haemophilus influenza. All these organisms are involved in the disease meningitis, and it is very important for us to establish the organism or the causative agent which is involved in the disease causation. So once identified, it helps in the treatment of that particular disease. So establishing cause is very important, because meningitis can be caused by different group of organisms. So once established that it is caused by the Neisseria meningitis, that is a purulent meningitis, then we can go with the further lab diagnosis. Here the lab diagnosis can be done with the specimen collected from the cerebrospinal fluid or the blood. And the carriers, in case of carriers, because this organism cannot cause the infection in them, the organism is present only in the nasopharynx, and with the help of pharyngeal swabs, you can identify the organism or inoculate the organism onto the medium. Now coming to the specimen, that is a cerebrospinal fluid. Here in the case of the patient, the cerebrospinal fluid is under pressure and it becomes turbid with more amount of the pus cells. So if we have a large quantity of specimen, we can divide the specimen into 3 parts and can carry out various laboratory technique to identify this particular group of organism that is involved in the disease causation. So here in this particular technique, the cerebrospinal fluid is divided into 3 parts. First part of it is used for centrifugation, and it is centrifuged while after centrifugation you can see a deposit and a supernatant. In the deposit you can go with the gram staining technique. In the gram staining, identification of the diplococcus with flattened adjacent flattened surfaces helps in identification of the organism. That is the initial presumptive test, you can say. Once it has been diagnosed that the gram negative organisms are there with the flattened adjacent site, then you can go with the treatment. Immediately treatment can be started. Next is coming to the supernatant. Supernatant is mostly used because to identify the antigens. So in the supernatant you can find the antigens that are related to the organism. So antigen can be identified either with the latest agglutination test of chemo or counter immunoelectrophoresis. Now similar type of test can be done for the other group of organism. As we know that other group of organism are also involved in meningococcal infections, you can go with the antigenic determinants or antigenic identification of which group of organism is causing that particular disease. Next part of the cerebrospinal fluid is inoculating onto the culture media. You can go for blood agar or the chocolate agar. Once the growth of the organism is there, you have to group because there are different kinds of Neisseria organisms so involved in meningitis. So you have to group them in order to identify the main causative agent, and grouping is usually done by agglutination or other serological techniques. And the final part of the cerebrospinal fluid is either directly inoculated onto the media or it can be incubated overnight with the glucose broth, and further on subculturing they can be done on the chocolate agar. So these are the different techniques by which we can identify the organism in the cerebrospinal fluid. Next we can go with the blood culture. Here the organism can satisfactorily grow in a blood culture, but they will not show hemolysis. Weak hemolysis is seen, but the antigenic components can be seen in the blood culture because of the bacteremia. And here the organisms are culture for 4 to 7 days. Immediately every day you have to go for the subculture onto the media to identify the organism or growth of the organism. In case of carriers who carries the organism in the nasopharynx, in those person you can go for the nasopharyngeal swabs. The carriers are the one that carries this organism and are able to transmit to the other persons. So we are collecting the swabs or while collecting the organism, aseptic techniques have to be followed because there might be a contamination with the saliva that can get contaminated with the normal flora of the mouth and cause a confusion in the diagnosis. So most probably the nasopharyngeal swab have to be collected with aseptic technique, and immediately it has to be transported to the transport medium, that is a Schwartz medium. Next, coming to the petechial region, which is a characteristic feature of this organism. Here you can directly isolate the organism or from the microscopic technique, that is by making a smear of it and identify the gram nature of the organism, or you can go for the culture in various media, that is a blood agar, or you can go for the Muller-Hilton agar or modified Thayer medium. Next is the autopsy. This organism, because they are involved in the meningeal infections, they can be isolated from the meninges, lateral ventricle, surface of the brain, spinal cord, and they have to be immediately identified within 12 hours before the death, after the death of the patient. Now coming to the retrospective evidence, here we can go with the antibodies, determination of the antibodies, because these antigenic substances will have the ability to produce the antibodies. Now the identification of the antibodies with the various serological techniques, you can know the causative agent involved in the disease causation. Next you can go with the molecular diagnosis for groups specific organisms that are involved in the disease causation. So for this sake you can go for the DNA sequencing or PCR technique, that is a polymerase chain reaction. Coming to the treatment, the organism can be easily treated with the help of sulfonamide in the early stages. That is, it was found that sulfonamides were effective on this organism at the early stage, but later on this organism has developed resistance. So we can shift the treatment from the sulfonamides to intravenous penicillin G. Nowadays we have found that this organism also have the ability to develop resistance against the penicillin. So can move on to chloramphenicol treatment or cephalosporins. That is a third generation cephalosporins can be used to treat these infections. Even before we find that the person is suffering from meningitis, you can go with the treatment with cephalosporins. And for the eradicative therapy, you have to use rifampicin, because making use of cephalosporin penicillin, you cannot eliminate the organism. You have to completely eradicate in the case of carriers. So in those patients we can, in those case of carriers, we can make use of rifampicin or ciprofloxacin. Coming to the prophylaxis, these organisms, due to its resistance towards different types of antibiotics, you can go for the chemoprophylaxis, making use of rifampicin and other ciprofloxacin. And now, moreover, we have a monovalent or a polyvalent vaccine against this organism, against the capsular polysaccharide, which is highly virulent in nature. So, the organism towards which we can usually protect ourselves with the vaccines are belonging to the group C, A, C, W, 135 and Y, which are mostly involved in the disease causation. And it is a single dose of vaccination is effective in case of older children and in adults, but they do not provide immunity to the children who are less than 3 years of age. So this is about Neisseria meningitis.